Welcome to the PubMed Daily Review for April 19th, 2026. Today we scanned 3,059 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5. Junction Opener enables CAR T-cell treatment of solid tumors by Stephen J. Reed and Derek Carter from HDT Bio, 1150, Eastlake Avenue East, Suite 200A, Seattle, Washington, 98109 USA. Let's break down its key contributions. Chimeric antigen receptor, CAR, T-cell therapy has been a game-changer for blood cancers. However, its success in solid tumors has been limited. This is largely due to the physical barriers within solid tumors, such as tight junctions between cells, that prevent CAR T-cells from effectively infiltrating and attacking the cancer. This research investigates a novel technology designed to open these junctions and improve CAR T-cell access. To test their hypothesis, the researchers used both in vitro tumor spheroids and in vivo mouse models of solid tumors. They employed a novel protein called Junction Opener, or JO4, which targets a molecule called DSG2 on cancer cells. The idea is that JO4 temporarily opens the tight junctions between tumor cells, allowing CRT cells, specifically those targeting HER2, to infiltrate the tumor more effectively. They then assess CRT cell infiltration, tumor disruption, and overall tumor control. The results were very promising. In vitro, JO4 treatment led to a significant increase in CAR T-cell infiltration into tumor spheroids and a dose-dependent enhancement of tumor cell killing. In vivo, mice treated with both JO4 and CAR T-cells showed significantly prolonged tumor control and improved median survival compared to controls. These findings strongly suggest that JO4 effectively facilitates CAR T-cell therapy for solid tumors. This study demonstrates a compelling new strategy to overcome a major hurdle in CAR T-cell therapy for solid tumors. By opening physical barriers, the junction opener technology could potentially allow for lower CAR T-cell doses, reduce manufacturing burdens, and improve treatment outcomes. While these results are exciting, further research in human models is crucial to validate these findings and pave the way for clinical application. Up next, our second paper out of 5. Patient-Derived 3D bioprinted intrahepatic cholangiocarcinoma models recapitulate tumor autologous traits and predict personalized adjuvant therapy by use Liu and Eli Mao from Department of Liver Surgery. Peking Union Medical College, PUMC, Hospital, Peking Union Medical College, PUMC, and Chinese Academy of Medical Sciences, CAMS, Beijing, China. Let's break down its key contributions. Intrahepatic cholangiocarcinoma, or ICC, is a devastating cancer with a very poor prognosis. A major challenge is that every patient's tumor is unique, making it hard for standard treatments to work effectively. This study aimed to create a new way to test drugs on a patient's own tumor cells outside the body, hoping to find the best treatment for each individual. The researchers developed a novel 3D bioprinting technique using a special hydrogel to create models that mimic the patient's actual tumor environment. They used cells from 21 ICC patients. These bioprinted models were then used to test the effectiveness of various cancer drugs, aiming to see if they could predict which treatments would work best for each patient. The results were very promising. The bioprinted models closely resembled the original tumors in terms of structure and genetic makeup. Crucially, the drug responses seen in the models perfectly matched what happened to patients who received those drugs. This means the platform could accurately predict which therapies would be effective, and it could do so quickly within just 10 days. This study demonstrates a significant advancement in personalized cancer care for ICC. The 3D bioprinted models offer a rapid and reliable way to test drug sensitivities, potentially guiding clinicians to the most effective treatments for individual patients. While promising, further studies are needed to confirm these findings in larger patient groups before this technology can be widely adopted in the clinic. Up next, our third paper out of 5. APO4 drives microglial lipid dysregulation in Alzheimer's disease via epigenetic reprogramming of the ASCL1/LXR-alpha-H3K4Me3 axis by Lanyan Lin and Fan Lin from Geriatric Medicine. Fujian Key Laboratory of Geriatrics Diseases. Fujian Provincial Center for Geriatrics. Fuzhou University School of Medicine. Fuzhou University-affiliated provincial hospital, Shunli Clinical Medical College of Fujian Medical University, Fuzhou, China. Let's break down its key contributions. Alzheimer's disease is a devastating condition, and the APO4 gene variant significantly increases your risk. We know that microglia, the brain's immune cells, can become overloaded with lipids in AD. But how exactly does APO4 cause this lipid buildup, and what are the consequences? This study aimed to uncover the molecular pathway connecting APO4 to microglial lipid problems. To investigate this, the researchers used a sophisticated approach. They studied mice engineered to carry either the human APO3 or APO4 gene. Using advanced techniques like single-nucleus RNA sequencing and epigenetic analysis, they examined microglial function. They also manipulated gene expression directly in microglia and tested how well these cells could clear amyloid beta, a hallmark of AD. The results were striking. In mice with the APO4 gene, microglia showed a significant, age-dependent increase in lipid droplet accumulation, up to 3.5 times more by 15 months compared to APO3 mice. This lipid overload was linked to impaired function, as these microglia were much less efficient at clearing amyloid beta. Importantly, the study found that APO4 led to reduced expression of a key gene called ASIL1 in microglia. When they experimentally boosted ASIL1 levels, they were able to restore normal lipid processing and improve amyloid beta clearance. So, what does this mean for Alzheimer's disease? This study reveals a specific epigenetic pathway through which APO4 contributes to AD by disrupting microglial lipid metabolism. The gene ASIL1 emerges as a key player in this process. These findings open up exciting new possibilities for developing therapies that target ASIL1 to protect against APO4-driven Alzheimer's pathology. Up next, our fourth paper out of 5, CD133, Guided RNA Nanoparticle Delivery of FTO-siRNA Impairs Leukemia Resistance to Tyrosine Kinase Inhibitor Therapy by Huiqin Bian and Shujun Liu from Department of Medicine. The Metro Health System, Case Western Reserve University, 2500 Metro Health Drive, Cleveland, Ohio, 44109, USA. Let's break down its key contributions. Leukemia treatment has advanced significantly with tyrosine kinase inhibitors, but drug resistance remains a major hurdle, leading to relapse and the need for lifelong, toxic treatments. Targeting therapies specifically to cancer cells is also difficult. This study explores a novel approach using RNA nanoparticles to deliver a gene-silencing agent to overcome this resistance, focusing on a protein called FTO, which appears to play a key role in making leukemia cells resistant to treatment. To tackle drug resistance, the researchers developed a sophisticated delivery system. They engineered RNA nanoparticles, which are like tiny delivery vehicles, and decorated them with a targeting molecule that binds to CD133, a marker found on resistant leukemia cells. These nanoparticles were loaded with FTO-siRNA, a molecule designed to silence the FTO gene. They tested this system in lab-grown leukemia cells, including those resistant to tyrosine kinase inhibitors, to see if it could effectively deliver the FTO-siRNA and reduce FTO expression. The results were very promising. The CD133 guided RNA nanoparticles were efficiently taken up by the resistant leukemia cells. Crucially, this targeted delivery led to a significant knockdown of FTO expression within these cells. This reduction in FTO was associated with a notable impairment in the formation of colonies and spheroids, suggesting that silencing FTO can indeed help overcome drug resistance and hinder the growth of these resistant leukemia cells. This study highlights a significant advancement in developing targeted therapies for leukemia. By using RNA nanoparticles to deliver FTO-siRNA, researchers have demonstrated a promising strategy to overcome drug resistance. This approach not only targets the FTO gene, but also specifically targets the resistant leukemia cells, potentially reducing side effects. While these findings are exciting, further research, particularly in animal models, is needed to confirm these results and assess the safety and efficacy of this novel therapeutic strategy in a clinical setting. Up next, our fifth paper out of 5. Dysregulated lactate metabolism synergizes with ALS genetic risk factors to accelerate motor decline by Shweta Tendulkar and Aaron D'Antonio from Department of Developmental Biology, Washington University School of Medicine, St. Louis, Missouri, United States of America. Let's break down its key contributions. Neurons depend on glial cells for energy through a process called lactate shuttling. This vital support system declines with age and in neurodegenerative conditions like ALS. While complete failure leads to axon breakdown, we wanted to know what happens when this system is only partially impaired. This research explores how problems with lactate metabolism, specifically involving the LDHB enzyme, might make the motor system more vulnerable to damage and disease. To investigate this, the researchers used mouse models. They studied mice lacking the LDHB enzyme entirely, and even those with just one working copy, finding significant motor impairments. They also pinpointed that the problem lies in Schwann cells, which support nerve function, rather than motor neurons themselves. Furthermore, they examined if this metabolic issue could worsen the effects of known ALS genetic mutations. The results were striking. Even a partial loss of LDHB function, as seen in heterozygous mice, significantly impacted motor behavior. When LDHB was specifically removed from Schwann cells, it led to substantial motor problems. Most importantly, when this metabolic deficiency was combined with common ALS genetic risk factors, it dramatically accelerated the onset of motor neuropathy, showing a clear synergistic effect. This study reveals that lactate metabolism is a key player in motor system vulnerability, not just a passive support system. It highlights that impaired LDHB function, especially in the supporting Schwann cells, can directly contribute to neurodegenerative processes. These findings open up exciting new avenues for therapy, suggesting that manipulating lactate metabolism could be a promising strategy to slow or prevent neurodegeneration in conditions like ALS. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.